Welcome to our next seminar organized by the Center for AI Innovation at NCSA. Today, we are very happy to have our next presenter in our series, Weihao Ge, who is a research scientist at NCSA in the industrial applications domain. She has a PhD in biophysics, and she has a lot of experience in analyzing statistical models for biological systems, from gene ontology, enrichment analysis to epidemiology models. And so the work that she's going to talk today about is going to be how her group analyzed surveys data and test data on the UIUC campus with regards to COVID, and how this data was then instrumental in helping the university administration in making decision policies about safety measures and activity guidelines and so on. So Weihao, you're welcome.
Thank you. Yeah, so the basic theme today will be telling you the reason behind the university policies and why our life makes so hard due to these health policies. Because these health policies are derived based on analysis on the survey and the test, and we found out that we have to use these health policies to keep everyone safe. So I will briefly talk about the background about the campus-wide testing, the monthly survey, and the SHIELD project, because our project is a part of this big SHIELD project against the COVID to ensure our UIUC campus is safe. And I will talk about how do we do the data pre-processing and some descriptive statistics and survival analysis. And also, I will present some results that, based on these results, we formed some conclusions that are helpful for the campus admins. Okay, so the background is that I believe everybody has participated in the cellular-based test and answered monthly surveys. And these parts has formed the SHIELD project, which involves a lot of different parts, for example, to collect the test data, to process the test data, to securely transport the data and store them and merge them with the information in the banner system, and also merge them with the monthly survey. And our group's work is on the very downstream of this project, that we will analyze these data to build a model to tell everyone that what do we derive from all these data. And what we get is from the de-identified information. And we work it on the Nightingale computer cluster, which is a HIPAA compliant system. That is, the cluster is designed specifically for securely handle the human information and the medical health record. And we also obtained the consent for research from some of the subjects involved. That means the consent of the research from some of the students, faculty, and the staff that if they are willing to give their test result and survey result for the research. And also we use the data to do the risk assessment for the fall 2020 semester. And also the risk assessment is based on the, since it's not for the research publication purpose, it's for the administrative purpose. So we use the full data set instead of the data set that's only consented for the research. So the results I show here, except for a publicly available figure, all of these are based on the consent for research data subset. So it will be like less representative than the full data set. And we have a very strong collaborative team, including the team from NCSA Genomics, which worked with Professor Rebecca Smith, who is also here today. And we worked with her on the West Nile virus epidemiology analysis in the Chicago area. And later, we also moved on to analyze the COVID data. And we also have more research scientists research programmers from the NCSA genomics groups and data scientists from the campus. And before we get the data, there will be a lot of processing and paperwork to do. So what we did is to create a synthetic data set for the code development to make the model. And also we use the synthetic data sets to validate our model to make sure that when the real data comes, we already have a valid model to work with. And once we got the real raw data set, we created a scheme to clean the raw real data set. For example, we use it to remove the outliers, to input the missing values, and to remove the inconsistencies. And then we started to create the codes to pre-process the data and also merge the multi-month data set. For this code, we make it very easy. It's like a one-click module for us so that in the later months, we do not have to spend a lot of time worrying about how to run the pre-processing code. And then we improve our model over our biweekly discussion with Professor Rebecca Smith. And then we polish and complete the code set for the survival model. Afterwards, we done more analysis in addition to the survival model. So that's why today, although the theme is about the survival model, which we spend most of the time working on it, but the represented results might include a lot of other analysis in addition to the survival model. So what is the survival model? The key question for the survival analysis is, what is the expected duration of the time until an event occurs? So here we will be asking, what is the expected number of days until a person can be tested positive? And to build this model, we also made some assumptions about our data. First, the people have consistent identities over the semester because their gender and race are self-reported. So we will assume that their identities does not change. Otherwise, it will be a very rare case, so it will be very hard for us to analyze. And we also made a strong assumption that their behavior is consistent or averaged over each month, that we are assuming that they have the same risks each day in each month. And we are assuming that the behaviors have immediate impact on the risk. So that means on the first day of each month, their behavior change and their risk immediately change. So if that person got tested positive on that day, we will assume it's the behavior in that month, not the behavior in the previous months. And also, the survey describes the behavior of the previous months. That means what we defined is the August data will be the September survey plus the August test results. And also, the first day tested positive is the first day being positive. And thanks to the frequent testing, it's not a very bad approximation, actually. So then we start, based on these assumptions, we are collecting the variables we need to pre-process and reformat the data so that we can put it in the model. First is the test data that we get the test timestamp and the test result. So that means if a subject takes a test on what time on what day and they got a positive or negative, that will be the result we receive. And we will convert it to censored or not. That means if the subject has all negative tests during a month, or do they have a positive test during that month? And we will calculate the so-called survival days. That means that's the days a subject stayed negative before the first positive test result is come out. And in addition to these two dependent variables for the survival analysis, we also have the test frequency, which is a variable we are interested to investigate the risk of taking tests. Because if you're going to take tests, you have to go to the booth and you have to be interacting with people and you have to take off the mask to collect the samples. So some people are very concerned about if taking tests themselves has bringing the risk. So we use the test of frequencies to show if it's risky or not. Theoretically, if the test itself brings more risk, then the higher the frequency they take the test, the less likely they have to, they could stay negative. That means they will have a shorter expected survival day. And we also retrieved the demographics data from the Banner system. And there are a lot of categories from the Banner system. And for the ones we take the rows and birth years, because for the other categories like the campus and well, I don't remember the other parts, but they kind of have too many categories that we have very, very few people in each category. But for the row categories, we also have to merge some of the overlapping row categories. For example, we merged the law school students to the graduate students, and we merged the two different categories of emeritus faculties into one category of emeritus faculties. And we convert the birth years into ages and merge them into the age groups. And then we pre-process the survey data. So first we remove the inconsistency in the identity data. That means if an individual thinks themselves as female for the most of the months, and in the other months they think they are male, we will just assume the person stays female all over the semester. And for the missing value in the behavior data, we will see that if a person has a missing value, they did not answer the question in the survey. So we might want to see what is the most people's answer to that question and assign a value. And for the outliers, we observe some students writing that they have the over 9 million people in the social bubble. So we will we draw a box plot and finding an upper cutoff at about 300 people. So we will impute any number larger than 300 people with 300 people. And for the housing type, there are a lot of people who does not answer, they choose the other. So we look at the data, look at the specific answers of the other, and find out that actually they could be manually categorized into the existing housing types. So we did this manual imputation. And also we did some stratification to merge the continuous variables into levels. For example, how many people are sharing a dwelling? We changed that to maybe no people sharing a dwelling, like 2 people, and then we use some quantile to see if they have like 4 people or more than 15 people. And also for the test of frequency, we merge that into levels of less than 1 test per week, and 1 to 2 tests per week, and more than 2 tests per week. So first, we want to perform some univariant analysis on the consistent variables that we want to know what groups of individuals are more likely to be affected. So based on our previous experience, if the variable does not vary a lot over the time, then the signals will tend to be masked by the signals of the more higher varied variables. So in our case, the identities and their roles, their badges, their age might be these variables, and their signals might be masked by the monthly changed variables. So we perform the chi-square test for the mutually exclusive categories. So first, I want to look at the representativeness of the survey. that among the 5,222 subjects, we performed over 100,000 tests, and 167 positive individuals, 167 positive subjects are identified. And among those subjects, 3,948 responded to the survey. But sometimes the survey and the test does not align very well. For example, the person took a survey this month, but they didn't take the test that month. So among the people who has matching survey and tests, we have 77 positive people out of 3,859 people. So we have found that we have very significant p-value showing that the survey with the research consent is not representative, that it's too optimistic, that the people who answered the survey and the people who gave the research consent are less likely to be positive. And we also look at the batch type. So for the batch type, we then analyzed the positive numbers, and we further found that the positive numbers outside the undergrads are we have too few numbers, so we have to merge them for the analysis. So we identified that 104 out of the 1,742 undergrads who has provided the research consent are positive, while for the other people we have 62 people, and we found out that undergraduate students are more affected by the COVID-19 significantly. And for the age group, first we try to stratify the age group by the risky group and versus non-risky group, but the risky group have too few positives. So we have to split the age groups into the quantiles. So each quantile approximately have the same number of population. And we found the youngest group are significantly more affected. That's consistent with the undergraduate student analysis. Then for the gender, we dropped the non-binary, self-described, and no answers because they have too few positives. And we found out that the female students consented for the research are slightly more affected by COVID-19, but still it's significant. And for the race, I cannot get any meaningful results that except for white European Americans, other categories have too few positives for the analysis. And for the other categories, since they are highly heterogeneous, I don't think it's like reasonable to merge them like the previous times, like the previous cases. So I have to say that for the race, we have too little information to draw any conclusion. And for the housing type, again, we have met with the analysis issues that we have too few positives. But we found out that the housing type is correlated with the batch type, that the undergraduate students, their housing type components are very different from the other groups. So we will still think that, okay, we will just use the undergraduate students' features as the feature in our model to analyze the risk of the population. And we will also emphasize on delivery the information to the undergraduate students to reduce the COVID risk on campus. And then we will do more of the Kaplan-Meier analysis, that we want to know that how the survival time is affected by certain factors. Those factors might be changed over the month, over monthly. And for those factors, we cannot stratify the population mutually exclusively, because a person can behave differently over the different months. And the questions we are interested here mostly are how safe are the in-person classes and labs, and how safe are the tests themselves. So we will present the results in the figure of the survival probability change with time, that on the x-axis is all days, and on the y-axis is all the survival probabilities. And the survival probability will decrease with time because it's the evolvement of a poisson process. And the faster decrease means that activity has higher risk. And we also include the uncertainty range to show whether the difference of the risks are discernible. So first is in-person class lab and safety. So I have verified multiple times that these curves are not wrong. Based on our calculation, the people attending the in-person class or lab actually have a better, although not very significant, survival probability than the people who are not attending the in-person class or labs. That means our in-person classes and labs are safe. We cannot simply conclude that it is safe to go to class or go to class make you safe, but it means that the measures the campus has taken to ensure that in-person instruction and research are actually effective, that the people who go to the in-person class or lab are safe. And the same thing happened to the frequent test safety. And we have found that mostly the range of the uncertainty is overlapping with each other, so we cannot really discern whether the people are having less or more risks. But based on the average value of the curve, we can see that people who take more tests do not have more risk or less survival probability than the people who take less tests. So that means it is safe to continue enforcing the tests. And then we made the Cox Regression module to mathematically calculate the hazard ratio. That is, how is the risk increase if an activity or safety measurement is taken comparing to not taken. The Cox regression is mathematically equivalent to the Kaplan-Meier analysis, but the result is represented in the hazard ratio. That, for example, that means if I go to a bar frequently versus if I don't go to the bar, how many folds more likely I'm going to get COVID? Or if I wear a mask versus I don't wear a mask, how many folds less likely I'm going to get COVID? So for the research consent data, we identified no significant result. But based on the full survey data set, we have made this publicly accessible graph showing the risks of activities where the students can get reference to and evaluate their own risk before they take actions. And for these risks, for these activities, they are kind of different from what we answered, the questions we answered in the survey. So what we did is to map the risks onto the different categories. And we also discern if, for example, if a person go to gym with or without the mask, we simply multiply the hazard ratio to calculate if it's helpful to take the safety measurement if you want to take a relatively risky activity. And then we categorize the risks based on their rank and also based on their own values. The safe measures will always have a hazard ratio that's less than or equal to 1. That means you do not add your risk. And then we were analyzing for the vaccine policies when the vaccines are developed. So we will want to know that, do we have to enforce the mandatory vaccination? And can we reduce some safety measure enforcement when people get vaccines? So the questions here is, how are people's vaccination intentions related to their conformity to the safety measures? Are the people less likely to take the vaccines, also less likely to conform with the safety measures? And what we found is that we will use the concern level as the intermediate variable instead of the vaccination intention itself, because the reason to reduce the people's vaccination intention is either they are not concerned with COVID or they have a bigger concern with the vaccine. So first, we plotted the bar graph to see if the people who are less concerned are less likely to take the vaccine. And we got the answer, yes. The people less concerned are less likely to get vaccines. So that means concern level can be a good indicator of people's intention to take vaccine. And we also compared plotting the concern level versus plotting directly the vaccine intention. That for the people who are who are not at all concerned, we found that they are more likely to socialize with others with no safety measurement. And for the people who are very unlikely to take the vaccine, there will be a group of people rather stay by themselves to keep themselves safe. But there will be also a group of people who like to socialize with others. And the concern level describes this population better. And also notably that the people who are both a little concerned or somewhat unlikely to take the vaccine, they have approximately the same proportion of the people who do not want to take any social safety measurement when they are socializing with a lot of people. So that means this population is also very dangerous. And what else can we find is that the social bubble is, the COVID bubble is almost very rigid for each group. That means it's not for a person's choice that they keep a COVID bubble, but they have to keep a COVID bubble maybe due to the nature of their work or due to the nature of their family or due to the nature that they have to take the in-person classes or do the research in person. So the COVID bubble part is kind of rigid. So I then plot the concern level versus other safety measurements. The people with the less concern level tend to ignore the social distancing. And they also tend to wear a mask less often. And also they tend to clean their hands less often. So as a conclusion that we have to enforce mandatory vaccination because those who are less likely to take the vaccine are also less likely to keep conformed with the safety measurements. So that means if we do not have the mandatory vaccination enforced, we will have a very big pool of the COVID incubator running around in campus. So our work has emphasized the necessity to communicate with the undergraduate students because they are mostly affected. And we have confirmed that the in-person class lab safety measurements are sufficient. And we have confirmed that it is safe to take the tests. And we have identified the risky behavior and sent it to the campus. So before the vaccine was widely available, that the students can know which kind of behaviors to avoid. And we have demonstrated the necessity to enforce the mandatory vaccination. So our team includes Dr. Christina Flick, who worked to organize the team and also negotiate with the different parts of the campus to get us access to the data and get us access to the cluster and everything. And also me leading the science parts of it. And we have Dan Lanier, who is now retired, who was a former member of the NCSA genomics team, who performed the most of the coding part in the pre-processing code. We have Dr. Dufan, who has now moved to Google, and he worked with most of the pre-processing scheme and helped with develop both parts of the team, both parts of the code, and help to sync the codes together and debug the code. And we also have Curtis Donaldson helped us to also get access to the data. And we had a very talented undergraduate student, Wang Wei, who is now a master's student in Harvard, and he developed the most of the modeling parts. And our greatest acknowledgement to Professor Rebecca Lee Smith, who has instructed us over this project, providing very helpful insights and help us to get the results delivered to the administrations. And we thank the SHIELD project for supporting the project. And we will thank the Nightingale cluster to provide the computation system and also the database to hold the data. Thank you.
Well, thank you very much for your presentation. This is really very insightful into all this process. So if anybody has any questions, this is the time to speak up. Please, Rebecca.
Just applauding.
Yeah.
Thank you.
You know, to get started, I do have one question. So some of these results that sort of indicates that going to the class and taking tests more frequently, that indicates that it's kind of a safe behavior. To what degree actually you think this is sort of a self-selected group of people that are already very conscious of what they are doing anyway?
Yes, this could be a very good reason that going to the class is and taking the tests are related to a more safe population. Yes, this could be the reason. I agree. Yeah, and this will be very one of the very strong explanation of that. And the behavior themselves might not be safe because people have the exposure to other people, but at least we together worked to make it safe.
I see.
Yeah, and also if our measurement has reflected that there's risky, since at that time we have bi-weekly meeting with Professor Smith, so we will immediately reflect that finding to the campus, and the campus will do the adjustment, but that did not happen.
I see. So I guess, you know, so the main finding here is really that you kind of cannot quite rely on students doing the right thing unless you tell them what that right thing is. Yeah,
yeah, exactly.
Is this any different or to what degree it is different, if it is different to graduate students versus undergraduate students?
Yeah, the graduate students' behavior is more like the faculty and staff, and the undergraduate students are especially risky.
I see. So this is you can clearly cluster this undergraduate students and then everybody else in a different category.
Right.
I see. So have you thought about applying any other sort of machine learning models to try to extract some other insights from this? Or
yeah, actually, before Dr. Dufan left to Google, he has left a very good code set for the Bayesian analysis, and we are still running the code set and working on the analysis of that.
So are there any specific questions that you would like to be able to answer with this other types of analysis like Bayesian?
We will want to extract like how does the risk evolve over the months?
I see.
Yeah. So currently we are treating the subjects as a new subject in a new month.
I see. I also wonder, you know, what sort of connection there is there with some sort of social media that students are connected with as well. You know, if somebody says, "Hey, don't worry about wearing masks," is it possible to trace it somehow to more students all suddenly stopping wearing masks or something like that? I mean, so is there some sort of influence of the social media on the behavior of the students? I guess, what would it take to analyze that sort of connection?
Yeah, so I guess that will need some like web crawling
Yeah, right.
proficiencies and do the semantic analysis, which I believe previously we had a student, but unfortunately she has quit the team due to some emergencies. But she has experienced in analyzed Twitter posts about the positive and the negative emotions in the words, and if she were on the team, then probably she will analyze some Twitter, analyze some tweets on the Twitter or something on the Facebook, and help us to cluster those findings.
Is any of this data available for others to try to do some analysis, or is it possible to generate some sort of anonymized data?
So for this data, I believe we need to ask some other people in the SHIELD project. But what we received is that we cannot spread this data, although we have anonymized and also merged this data.
There is a process by which you can apply for access to the data, and I will drop in the chat that the link to that process.
Yeah, so there is another question posted there. So with the return of the football season, is this one of the types of activities that is going to impact sort of the outcomes of distribution on campus?
Yeah, I believe so, because we currently are no longer working on this project. So I would be very interested if new data are available. But what I have read is that in Britain, the Oxford University has conducted a study after the soccer season actually in Britain, and that really aggravates the COVID spread and added new spike.
Interesting. But so this is not something that you are currently in your surveys, right?
Yeah.
Okay. That's interesting. Okay, well, so are there any other questions? Yes, there is a question somebody posted there. So what other information do you think would be possible to get, maybe not get, that would help in this sort of analysis?
Other information, let me see. So,
principally, if you think about it, now that you have done this experiment, what have you learned such that if you have to redo it again, what would you do differently? In particular, with collecting data.
So one of the things I'm very interested in is about the roommates' behavior, because many of the undergrad, well, it's not a statistics, but it's anecdotes that I heard many of the undergrads complaining that they got the COVID from their roommates, but there was no way for me to extract this from the survey data because we don't have any roommate. We're not allowed to have any information. But if there could be some graph networks created and anonymize these people and also reduce any of the exact address information of these people, then that can be helpful to help us better separate the groups. So that can help us also identify whether some activities are actually safe, but students kept all the safe activities, but they got the COVID from the roommate. That will blur the data a lot, I believe.
Okay, are there any other questions? Well,
actually, on the second thought, instead of roommate, we know that Apple has created long ago the contact information. So if the people in the survey could respond, if they get a lot of the alerts, that might also be helpful and easier to collect than the roommate information.
actually, so I remember going to another state, and all of a sudden my cell phone asked me, hey, in this state, we track close contacts. So
Yeah.
to enable this or something like that. And I guess we don't have that in Illinois. So that's kind of a database that I guess Apple potentially collects as well.
Right, I believe they did a decentralized alert system that is used Bluetooth to measure the close contact, and that's not on a centralized database. So if the people can self-report if they get a lot of alert or not, then that can already help us to stratify the population. And also the population are decoupled, so it's easier to analyze than the network, interpersonal network.
Yeah, you could potentially use it as a guidance to look into some specific groups or clusters.
Yeah.
Okay, well, thank you very much for this very insightful presentation and all this great work that has been done here. This is really very interesting and hopefully something that helps us to function normally as we keep going through this process. So thank you very much. Okay, so with that, we will conclude our seminar today. Thank you, everybody, for attending it. As I mentioned earlier, the recording will be posted eventually on our website. And so we are looking forward to another presentation next week. Thank you.
Thank you.